We move our bodies by contracting skeletal muscle. While the results of this action are plain to see, the mechanism behind the action is not. Contraction of skeletal muscle tissue occurs when actin and myosin myofilaments slide past one another, causing the sarcomere to shorten. It is important to note that the individual myofilaments do not change length themselves, but instead overlap each other to shorten the sarcomere. Moving in for a closer look, we can see the details of how this occurs. The molecules you see here are myosin, which at rest has both adenine diphosphate and phosphate molecules attached to the individual myosin heads, actin, tropomyosin, and troponin. When we decide to move, calcium is released from the sarcoplasmic reticulum. These molecules bind with the troponin molecules. This causes the tropomyosin molecules to move, revealing myosin attachment sites on the actin myofilaments. The single phosphate attached to the myosin heads gets released as the heads move to connect with the attachment sites on the actin. The resulting formation is called a cross bridge. The remaining adenine diphosphate molecule is then expended in what is called a power stroke, when the myosin heads pull the actin inward. Once the adenine diphosphate attachment is spent, adenine triphosphate molecules attach themselves to the myosin heads. These molecules trigger the release of the myosin heads from the actin attachment sites and are immediately broken down into adenine diphosphate and a single phosphate as the heads move back into their resting position. This is called the recovery stroke. If calcium is still present, the cycle will repeat again, and the sarcomere will continue to shorten until the calcium ions are transported back into the sarcoplasmic reticulum and the muscle relaxes.
Okay. Obviously, she was super excited about this. How many of you actually watched that video? And how many of this was the first time? Okay, some of you aren't voting, I get it, but maybe your hands are full of food or something. We're going to go through this mechanism today, and this video does a really nice job of explaining it step by step. And so after we go through it today, it would be advantageous, in my opinion, for you to come back to that and to watch that video again. Okay? Deal? So what I want to do is finish out where we were, and then we'll take a break and roll into the next lecture, which is kind of where that video picks up. But what you'll see today, much of today is going to be repeating this same information in numerous formats. Okay? That's the importance of today. Now, tonight we have a quiz that's, or a worksheet that's due, right? And then tomorrow night we have a quiz that's due. Many of you have finished that, but some of you are still working on it. Remember, save your work. You don't have to stay up till 11:59 to hit submit. You can just save your work and it'll submit automatically. Or if you're done, go ahead and just hit submit. And we have an exam on Wednesday. I had a couple of questions over the weekend as far as the schedule goes, so I just want to make sure that you guys understand where we're at. And like always, we'll get as far as we get, and then we'll stop for this exam, and some of this material might spill over into the next unit, okay? But we won't rush through. But that video kind of shows you what I want to talk about today. Now, the action potential and how it gets propagated. So if we back up to where we were, this is kind of where we left off. And hopefully you can appreciate now watching that video that the overlap that occurs, this is relaxed, now there's more overlap, the muscle's contracting, now there's a significant amount of overlap because those Z lines or Z discs are getting closer and closer together. This is an energy requiring process. We'll talk about later the cycling, like the video talked about, of the ATP, adenosine triphosphate, which is our main form of energy. But if we back up a little bit, we need to go to this neuromuscular junction. And if we look at the events that are happening at the neuromuscular junction, we're going to have, obviously, the movement of sodium and potassium with respect to an action potential. And it gets all the way down to the end of the axon. And at the end of the axon, now we actually open what we call voltage-sensitive calcium gates. Voltage-sensitive calcium gates at the end of the axon. That allows calcium, extracellular calcium, to come in. We're not at the muscle yet. Okay, we're not down here at the muscle site. We're on the presynaptic neuron. That calcium comes in, and it causes these vesicles containing acetylcholine to fuse with the membrane and dump their contents. Now acetylcholine floods into the cleft. It binds to the acetylcholine receptor on the other side, right? And it opens a channel. And it opens a channel allowing sodium to come in to the other side, which is the what? What are some names for this region right here? What's that? The postsynaptic side, or the muscle. What else? What's that? Motor end plate. Very good. That's actually what I was looking for. Motor end plate. Okay? Yeah, you should celebrate. You should give him a high five. Okay? Nothing wrong with that. Now, if sodium floods in on this motor end plate, what event has been propagated to the other side? An action potential. And how do you propagate the action potential down the membrane? What's that? So you allow for voltage-sensitive sodium gates to open up, and then sodium comes into the cell. It reaches threshold. You trigger an action potential. Remember that? We had an exam on that. Hopefully you remember it. Okay? So you take the action potential from this side, you convert it from an electrical signal to a chemical signal. Then you convert it back to another electrical signal. That's how you propagate the signal across the gap, or the synapse. All right, so now what happens on the other side? This is the stuff that was in the video that we're going to talk about in a couple of different formats. You guys know I like to say the same thing over and over again. At least that's what my kids tell me. I know, Dad, you already said that. Okay? Oh, I did? Well, why don't you clean your room again? Okay? So this is where we're going to be for much of today, the bulk of today. About 75% of today is going to be talking about this stuff. So the nerve is now communicating with the muscle. And it does this using action potential. See why it's important to cover the nerve first and action potentials before you ever get to the muscle? Well, we call this specific event excitation, contraction, coupling. Okay, here's an interesting tip. A lot of students are emailing me, I'm confused on motor neurons or motor units. I don't understand what they are. I don't understand this excitation, contraction, coupling. Did you know in your textbook there's something at the very back called an index? And you can look up these crazy terms, okay? And then it puts you a number next to it, and you can go read a little bit more detail about that topic. What was that question in the back? Okay. So, excitation, contraction, coupling is an important word for you to understand. You couple, you link excitement of the membrane to the contraction events of myosin and actin. Okay? Well, when you couple them, you have to use the neuromuscular junction, like what we just talked about. Here's that term, motor end plate. You have a synaptic cleft. You have acetylcholine. These are all the players that are involved. You also have acetylcholine esterase because after the contraction is done, or you've coupled the event, you want to be able to turn it off. You don't want it to be linked indefinitely, right? You don't want to be in this contractile state like all the time. Okay? So you want to be able to relax that muscle. So a relaxation is just as important in the contraction event. Because if you want to walk across the front of the classroom or run to your next class, if all you do is contract muscles, you'll get one step. That's it. And then we actually need to propagate the signal. So if you look at where we were, you see these little arrows that are coming down. The electrical signal comes down the membrane surface and it dives into the T-tubule network. And on either side of the T-tubule network are these terminal cisternae that are a component of what structure? Sarcoplasmic reticulum. And what's one of the purposes of the sarcoplasmic reticulum or the terminal cisternae? Store calcium, okay? So we're inside a cell now, but we're actually outside the cell because the signal is coming down the tube. That tube lumen is contiguous with the outside of the cell. So any voltage-sensitive sodium channels that are embedded in here are actually embedded in the membrane surface, just like what we're used to seeing. Well, the reason that this triad that we call the two terminal cisternae and down the center, the T-tubule, it's called the triad, is important is you get the electrical signal coming down the middle one, and then you have calcium being able to shuttle out from either side. As calcium shuttles out, you can see the little white spheres. The calcium releases. You blow up this figure down here. Calcium is going to find troponin and bind. That's what the video showed you. When calcium finds troponin, it links up to it and it causes a three-dimensional shift change. Okay? It changes its shape. As it changes its shape, because it's physically connected to tropomyosin, which is this long filamentous protein, colored kind of in this green tube color, it moves out, it shifts as well. So when calcium binds, troponin changes shape, it pulls on tropomyosin, and you can see this arrow, you see all these little black holes? Those are the little binding sites for the head of myosin, which is part of what filament? The thick filament. Okay? So calcium regulates whether or not actin, the binding site for myosin head is on the actin molecule. Calcium regulates whether or not that binding site is seen. Make sense? Okay. Well, next up, the myosin itself is going to attach to the myosin binding site. That goes in the first point. And where is that myosin binding site found? It's found on, it's on the thin filament, and specifically it's on what protein of the thin filament? Actin. Very good. If you're not following this line of questioning, then you're not ready for Wednesday. Okay? If I'm saying it too quickly, you're probably not ready for Wednesday. But by the end of the lecture, we'll have said it like 3 or 4 more times. So hopefully you'll start getting ready for Wednesday. Okay. This is the cycling that takes place. Okay? The cycling is going to have sort of 4 main phases. We have this myosin head that can actually cock back and forth. That's the actual physical motion that takes place. It's like this ratcheting motion that you saw in the video. Okay? We also know that it has to attach. It has to attach to that binding site that's found on the actin protein of the thin filament. See there, I said it a second time already. Right? The myosin binding site is found on the actin protein, which is part of the thin filament. What are the other proteins that make up the thin filament that you need to know about? Tropomyosin and troponin. Okay? So the thin filament, while it's thinner, it has 3 important proteins that make it up. And the thick filament is kind of boring. It just has these globular heads of myosin. Then we have this power stroke. We call it power stroke because it actually requires energy for this stroke to take place. That's the ATP. We're going to see systematically where the phosphates get cleaved and where the energy is liberated. But generally speaking, it's called a stroke because the head fires and strokes, and that slides the filaments across each other. And then you actually have to have reattachment, right? Reattachment of energy. So we'll get into more of these cycles throughout today, and you'll see them as we walk through. So you can see up here, this is the myosin head with ADP and PI. So there's that third phosphate. And you can see the position of the head. You see how the position of the head is cocked back? Well, then, this is with calcium bound. You can see the black myosin binding sites on the active protein of the thin filament. And this allows for myosin to find actin. And when it binds, that's called the cross bridge, when they actually link up. Okay? Then you move from this step to here, and you get what we call this power stroke. That power stroke liberates this energy that's been attached. It liberates this energy with this ADP, adenosine diphosphate, and the free phosphate molecule that's still available. That allows the firing from this cocked position to this forward position. Do you see that? Then, when it does that, you can kind of appreciate this blurry side over here. Some of you are like, how come it's blurry? Well, it's trying to infer motion, right? It's sliding from the left to the right. Then you move up to this arrow, and in order to detach, you actually have to bind a new ATP molecule. A new ATP molecule has to bind in order for this head to detach from the actin protein. And then you cleave a phosphate, and that takes it from the detached point to the cocked position. And this recycles again and again and again.
Where did you say the cross-bridge forms?
Cross-bridge forms right here. When myosin and actin actually interact. That's the cross-bridge. That's technically what the cross-bridge is. Again, if you forget, you have a couple resources. You can look back at the slides. Right? You can go to the YouTube lecture, re-listen to that little segment, or you could just simply go to your index in your textbook and go to that segment in the chapter and read about the cross-bridge. You could also Google it, I'm sure, but right, you've got unlimited. You guys are not in an era where you're limited on resources. In fact, the problem is there's probably too many. Okay, sifting through good information versus bad.
Does this have to use 2 ATP?
One complete cycle is 1 ATP. Behind you, sorry. He was just first, sorry.
So the cross bridge is formed, and then is it the ATP breaking off that breaks the cross bridge, or is it the ATP being added on that creates the breaking of the cross bridge?
No, this here, the cross bridge is still intact.
Yeah, so is it...
How does it happen here?
So is it the ATP joins and breaks the cross bridge?
Correct.
Or the ADP leaves?
No. This will stay connected until a new, fresh ATP molecule attaches. Then it'll release. Because what happens is, when the new ATP molecule attaches to the head, it causes a conformational change, and it lets go of the binding site. Evidence of this is, we'll see in just a couple slides, is rigor mortis. So the rigor complex is when your muscles get stiff after you're dead. Why? Because they never break these cross bridges. The moment you stop breathing, ATP availability plummets. So if there's no ATP, all of the muscles in your body will form what we call rigor complexes. We'll talk about it in just a second. Yeah.
I just wanted to know if this is like a good way of studying it. I'm kind of thinking of like rowboat almost, like, I don't know how.
Oh, with the oars?
Yeah, like
yes.
Okay, fine.
Did you see the video? Did you see the video when they took away some of the background detail? And it looked like these little things were swimming. That is the stroke, like of a row team. That's exactly a good analogy.
I just wanted to make sure so I wasn't studying this weird.
Nope, you're studying it non-weird. Okay.
Thanks.
All right. So if we continue on this theme, how does it relax? Right? So backing up, well, here it's connected, and then you're telling me ATP has to bind in order for it to let go. Absolutely. So relaxation is an ATP requiring step. An ATP requiring step. So one result of limited ATP or ATP depletion are muscle cramps. How many of you had a muscle cramp? Okay? Some of you, it hurts right now and it's like cramping. I know, okay? But that's actually neural tissue, not a muscle. But anyways. So a muscle cramp could be from a variety of different things, but one potential cause would be limited ATP availability. So it stays kind of, have you ever had like a calf cramp? Or your toes like curl up in your shoe and you're like, oh! And you're like, what is wrong with you? It's like, got a cramp, right? You're on the floor and you're like, holy, what? Wow, what a baby, right? You're like, no, seriously, it's like so bad. My toes are like curled up in this ball, you know, like, okay, right? But they're not, they're haters because they never had one. I get it, okay? So ATP availability is required for relaxation. All right, some of you are reminiscing, right? Okay. So, one second. So, when we relax, we need to remove the signal, which is the acetylcholine. So you want to get rid of it, destroy it. You want to re-sequester. That's a fancy word that means reuptake calcium. The way that you do that is you actively pump it back into the sarcoplasmic reticulum. If I say the word actively pump, what key feature would you be looking for, A, B, C, D, or E on the exam? Active. What would you be looking for? What would be significant about an active transporter? It needs what?
ATP.
ATP. Okay? So now I just told you have to have ATP to let go. You also have to have ATP to pump calcium back into the sarcoplasmic reticulum. That should make sense because an active transporter, careful, this is the cumulative portion of the class. An active transporter requires ATP. Why? It goes against the gradient. Where is calcium actually high? Inside the SR, and it's relatively low inside the cell, or outside the SR, right? Sarcoplasmic reticulum. So to pump it from the cytosol, right, or the inside of the cell, the sarcoplasm, you actually are pushing it into the SR, where it's actually a little bit higher in concentration. So you need active energy to run that pump. Inside the SR, you have basically a magnet for calcium known as a protein called calsequestrin. I actually really like this word because calsequestrin is a word that means calcium consumption. It sequesters calcium. So calcium has a high affinity for calsequestrin, and as it's pumped in, it's active, but once it gets inside, it kind of binds up to this molecule. This is an ATP-required process because of this active pump. So I've told you at least 2 locations or details of relaxation being an energy-requiring process. Now, if we look at the evidence, I know I've got a couple questions. If we look at the evidence of what happens if you don't have ATP available, this is where the Rigor complex comes in. Okay, so the Rigor complex, some details about it. This is stiffening or hardening of the muscles post-mortem after death. It begins as early as 3 hours. There's some evidence that it could occur a little bit faster. But 3 to 4 hours is the general window of time. What happens is the tissues start to decay, and as the sarcoplasmic reticulum decays, it just sort of dumps out its calcium. As soon as calcium is available, it binds to troponin, and tropomyosin moves out of the way, and the myosin heads bind to actin. That doesn't require energy. What requires energy is the movement of the head or the detachment of the head. So if the individual passes away and is in the anatomical position 3 to 4 hours later, that's the position the Rigor sets up in. If they're in some other position, that's the position that Rigor sets up in. Okay, so when you die, don't be like, doing something weird that you don't want everybody like, whoa. Okay. The deteriorating sarcolemma also allows calcium from outside the cell to come in. And this calcium availability allows for the muscle cross-bridge to form, but it can't let go because there's no ATP. It's no longer produced after death. The fibers remain stiff and contracted until those proteins start to deteriorate, and they will eventually, that linkage won't break. It'll be somewhere on the myosin head fractures off and deteriorates. Okay? So at about 12 hours after death is where it peaks, and then it kind of fades over the next 2 to 3 days. Okay, question?
I was just wondering how muscle cramps inside pain. When you get a muscle cramp, why do you feel that?
Why do we feel pain with your muscle cramp?
Yeah.
Well, because inside the muscle itself are pain receptors called nociceptors. And so they're being physically stimulated when the muscle balls up like that. And that's triggering a pain response. Okay?
I was going to ask, how do they help, like, dang it, what's that called? Like, after you die and you, like, donate your body to science and, like, they, like, dissect your body or
Cadavers?
Yeah, there you go.
Okay, yeah.
How did, I'm sorry, how did, like, they move their muscles if they're all, like, if they're stiff from?
Well, so with a lot of cadavers, you'll have limited movement. And depending upon how it's preserved, you may have better movement or not very much movement. So the way that we preserve tissues post-mortem for teaching and learning is actually with fixative. So you use a fixative, and that causes the proteins to cross-link in the native state that they're in. So if you cross-link the proteins in a relaxed state, then they won't ever link up. Okay?
During fixation and everything, when you release acetylcholine, and if you hold that, is that just not allowing the acetylcholinease to get in there and break it up, or you continually try to?
When you do what?
When you have that original linkage, when it comes down and it binds to release acetylcholine, does that acetylcholine stay until acetylcholinease shows up?
Yes.
Or do you keep firing more to replace?
Well, if acetylcholine stays in the cleft, you'll actually get repetitive contraction events. So usually what happens is acetylcholine release into the cleft, it binds, it's used up, and it's recycled. The next time you contract, it's new acetylcholine. If you hold contraction, then you will keep using that acetylcholine as long as it's bioavailable. And then as you recruit additional motor units, because let's say you're holding a load and now your arm's starting to shake, and as you're trying to maintain that, you will send additional nerve impulses. Every new nerve impulse sends new acetylcholine fresh. Okay? Okay. So a couple questions as we wrap up this segment, and then we're going to be doing this stuff again in the next lecture. What blank surrounds each muscle fasciculus, whereas blank surrounds each muscle fiber? What's the answer? B is the answer. So it's perimysium surrounds each muscle fasciculus, and the endomysium surrounds each muscle fiber. Okay, how about this one? Enlarged areas of the sarcoplasmic reticulum near the T-tubules are called what? Sarcolemma, sarcomeres, terminal cisternae, troponin molecules, or tropomyosin molecules. C, terminal cisternae. Very good. Questions?